Welcome back. Let's continue with Grade 9 exploration. New curriculum chapter 2. Cell, the building block of life. You started out as just one single cell. Today, you are a walking, talking universe made of over 30 trillion. But how did one cell become trillions? Welcome to the greatest magic trick in biology, cell division. Zoom in to the microscopic frontier of an onion root tip and witness the unstoppable engine of life, mitosis in action. It is a spectacular reminder that true monumental growth happens step by step, cell by cell, pushing endlessly forward to build something incredible.
Today, we will observe how cells divide in the growing root tip of an onion. This experiment helps us study different stages of cell division under a microscope. First, fill a glass jar with plain water. Place the onion bulb over the jar in such a way that its base just touches the water. After 5 to 6 days, new roots begin to grow from the onion bulb. These rapidly growing root tips contain actively dividing cells. Now, carefully cut 2 to 3 centimeters of the freshly grown root tips. These tips will be used to prepare the microscope slide. The root tips are placed in aceto alcohol solution for 24 hours. This preserves the cells and stops further cellular activity. After fixation, the root tips are transferred to 70% ethanol for preservation until they are ready for observation. The preserved roots are washed with water and placed carefully on a clean microscope slide. Dilute hydrochloric acid is added to soften the tissue, making it easier to spread the cells for microscopic observation. Next, acetocarmine stain is added. This stain colors the chromosomes, making the dividing cells clearly visible. The slide is gently warmed over a spirit lamp. Heating improves stain absorption and enhances the visibility of the cells. The tip portion of the root is covered with a cover slip and gently squashed to spread the cells into a thin layer. Now the prepared slide is observed under the microscope to study the cells present in the onion root tip. Under the microscope, different cells appear in different stages of cell division. These stages include interphase, prophase, metaphase, anaphase, and telophase. The cells of the onion root tip divide continuously for growth. This process is called cell division or mitosis and helps organisms grow and repair tissues.
Mitosis is the ultimate builder, perfectly cloning your cells to heal wounds and fuel your daily growth. But to create entirely new life, meiosis takes the stage, cleverly scrambling the genetic code to guarantee that every single generation is beautifully unique.
How do we grow and heal? Cells don't just get bigger, they continuously divide to replace themselves.
For normal growth and repair, cells use mitosis, the most common type of cell division. Mitosis produces 2 genetically identical daughter cells, ensuring your body's genetic information is perfectly maintained. For sexual reproduction, cells use meiosis. This process creates genetic diversity, making everyone unique. In meiosis, the parent cell divides twice, ultimately forming 4 unique daughter cells called gametes. Crucially, each gamete contains exactly half the number of chromosomes compared to the original parent cell.
Arun Kumar Sharma invented powerful new lab methods to study plant chromosomes, earning many honors, including the Padma Bhushan.
Time to pause and ponder. Why do cells have many small mitochondria instead of one giant one?
It's about surface area. Many small mitochondria provide a massively larger surface area to produce more energy. Question 2. What would happen to a cut if your skin cells started dividing by meiosis? It wouldn't heal properly. Meiosis halves the DNA, so the new skin wouldn't match the original tissue.
Bridging science and society. Scientists can now perfectly grow plant and animal cells outside the body.
This incredible process, called cell culture, is absolutely crucial for producing life-saving medicines and vaccines. Keep exploring.